Good afternoon, honorable scientists, distinguished guests, ladies and gentlemen. Welcome to the science seminar titled with this special theme, Kaleidoscope of Protein Folding. And today's seminar is co-organized by the Council of the Hong Kong Laureate Forum, namely the HKLF, together with the Hong Kong Science Museum. Allow me to introduce myself. My name is Tasha, and it's my absolute pleasure to be your master of ceremony today. Thank you. And we'd also like to take this opportunity to welcome everybody joining us in person today. And we'd also like to extend our heartiest welcome to our online participants joining us through the live webinar today. And of course, we have an excellent program today, but most importantly, we're very much honored to be joined by three distinguished scientists in life science and medicine, with two honorable Shaw laureates, Professor Franz Ulrich Hartl, and also Professor Kazutoshi Mori. Let's welcome our scientists with a big round of applause. And also together with a very promising scientist from Hong Kong, Professor Stephanie Ma. Professor Ma will be the moderator of today's seminar. She is the Professor and Associate Director of Knowledge Exchange and Global of the School of Biomedical Sciences in the Li Ka-shing Faculty of Medicine at the University of Hong Kong. She's also the founding member, Vice President, and Co-Chair of the Outreach Committee at the Hong Kong Young Academy of Sciences. Now, without further ado, let's put our hands together to welcome Professor Ma to come on stage to deliver the opening remarks and introduce the first speaker for us. Professor Ma, please.
Very good afternoon, and thank you, Tasha, for the very kind introduction. Welcome to the Hong Kong Laureate Forum Week, and of course to this science seminar on Kaleidoscope of protein folding. We're very blessed to be honored by many distinguished guests, laureates this afternoon. We have a very nice program put together. In particular, our two Shaw laureates, Professor Hartl and also Professor Mori, actually have common interests in proteins, one on protein folding, of course, and the other on unfolded protein response. So we'll be hearing from them, we'll be chatting with them, and we'll be happy to take questions from the audience. So I'll be acting as your moderator and host this afternoon, and I won't take any time away from you because you don't want to hear from me. Of course, you want to hear from the Shaw laureates. So our first speaker today is Professor Hartl. Professor Hartl is the Executive Director of the Max Planck Institute of Biochemistry, and of course, the 2012 Shaw Laureate in Life Science and Medicine for his pioneering work in chaperone-mediated protein folding. And his talk today will be on chaperone of the cells, role in protein folding and neurodegenerative diseases. So can I please invite you to join me in welcoming Professor Hartl onto stage, please? Thank you.
Thank you so much, Stephanie, for your kind remarks, and it's really a great pleasure and honor to be part of this forum, to be back in Hong Kong after 2012, and I'm looking forward especially to talking to so many young scientists and also some not so young scientists anymore. Yeah. Well, relatively early on in my scientific career, I was fortunate to be able to contribute to research that has changed our view of a fundamental process in biology, the protein folding process as it occurs in our cells. And I want to tell you today about this discovery and what it led to in terms of understanding how proteins are made in our cells. Proteins are, of course, the macromolecules that are responsible for almost any biological function. They are the most versatile molecules, and they perform the genetic program of the cell. Now, these are polymer chains of amino acid building blocks, and the important aspect here is that these chains, after synthesis, have to be folded, have to adopt a defined three-dimensional conformation. This is exactly what we understand by protein folding. Let me start out by explaining this with this historical slide. It shows David Phillips at the Royal Institution in London many years ago, presenting one of the first crystal structures of a protein. Crystallography, X-ray crystallography, is a method by which we can determine the three-dimensional structure of a protein. But the slide is important because it also shows you here the extended polypeptide, the extended protein chain as it is made on the ribosome machines of the cell, where the protein is in an unfolded state, in a linear chain, and the amino acid residue residue parts are exposed here at the side of this string. Now, here is a nicer version, perhaps, of a folded polypeptide chain. This is actually a rather complex protein already. It has multiple domains, multiple folding units, shown in blue and in gold. You can trace the polypeptide chain from the amino terminus to the C terminus. This is actually the protein that makes the light of the firefly, luciferase. We heard about luciferase this morning, different forms of this type of enzyme. Now, we have relatively powerful algorithms now, AlphaFold, for example, to predict the structures of proteins based on information that we have in the database, based on the information of protein structures that have been experimentally determined. We can now predict how a particular amino acid sequence will fold into a particular end state. What we don't understand so well yet is how the actual process of protein folding works, and that's what we are interested in, particularly from the perspective of the cellular biologist. Now, protein folding is the final step in the information transfer from gene to functional molecule, and as such, obviously, is a very fundamental process in biology. It is required for the vast majority of proteins to become biologically active in every type of cell. Many years ago, decades ago, in fact, 1950s, 1960s, Christian Anfinsen performed pioneering experiments that showed that you could artificially unfold a polypeptide chain, denature it, and then allow it to refold into its three-dimensional state by putting it back into a physiological buffer solution. And that demonstrated that the amino acid sequence of the protein chain contains sufficient information to specify the three-dimensional state, and that this folding process, importantly, can occur spontaneously without need for additional machinery. But even at the time of Anfinsen, it soon became clear that the spontaneous folding process works best only for relatively small proteins, perhaps up to 100 amino acids. But the majority of our proteins in the proteomes of mammalian cells have much larger polypeptide chains, hundreds of amino acids, thousands of amino acids. So how do these complex proteins fold that will not be able to do so in the test tube in the Anfinsen type reaction? In the late 1980s, people, including myself and my collaborator Arthur Horwich from Yale University, started to ask the question whether the process of protein folding in the cell is really a spontaneous reaction. Or are there machineries, like for every other major biological process, replication, transcription, translation, that mediate and assist in the protein folding reaction? We found ourselves in a situation where we had the opportunity to study this reaction, this folding process, using mitochondria as an experimental system. Here you see a mitochondrion. These are organelles of our cells where energy production occurs and various other metabolic reactions. So essentially tiny cells within the cell. And the important aspect here was that these proteins, these mitochondria, have to import most of their proteins from the cytosol after synthesis on cytosolic ribosomes, ribosomes being the machineries for protein translation. I was at the time a young group leader in the laboratory of the late Walter Neupert in Munich University, and we studied this process of mitochondrial protein import. We had realized at that time, the late 80s, that for protein translocation across the two mitochondrial membranes, the outer and inner mitochondrial membrane, the protein chain had to be in an unfolded state. So perhaps we could use this system then to study how a protein would fold up after its translocation into the interior space of the mitochondria. Together with Art Horwich, we had characterized a mutant strain of yeast in which a particular protein inside the mitochondria was defective, and many of the imported proteins were no longer able to form their final assembled states. And the critical experiment is shown here, where we used a monomeric model protein called DHFR and attached a mitochondrial signal for targeting to the mitochondria to its N-terminus, allowing its transport into the mitochondria, and we could then study its folding inside the mitochondrial space. Now, the DHFR was chosen because it is actually a protein that does fold spontaneously, just as Ann Finson had described in his test tube experiments decades before. But when we imported the protein into the mitochondria, what we found is that now the folding process became dependent on a pre-existent protein complex called HSP60. HSP stands for heat shock protein. And it required the input of metabolic energy in the form of ATP, the ATP molecules that are produced inside the mitochondria. Now, HSP60 is a molecular chaperone. It is now known as the mitochondrial chaperonin, a particular class of chaperones, and I will tell you in a few minutes in more detail how these machines work. But this experiment showed that the basic process of protein folding that can occur spontaneously in the test tube is no longer a spontaneous reaction under the conditions of the cell. And we now understand that the vast majority of proteins synthesized in cells require this type of machinery or similar types of molecular chaperones in order to fold efficiently and, that is important, at a biologically relevant time scale. And this machinery is not only necessary once when a protein first folds upon synthesis, a process that we call de novo folding, but proteins have to return to this machinery throughout their lifetime in the cell in order to maintain their functional conformations, a process that we call protein homeostasis or proteostasis. And you will later hear more from Professor Mori about particular aspects of how cells maintain this protein homeostasis, in particular under conditions of cell stress. And one aspect here is that under cell stress, cells augment the abundance of these molecular chaperone machines. Now, later it became clear that these chaperones also have implications in medicine in the context of various diseases where proteins no longer fold properly and form toxic protein aggregates, clumps of proteins. We know this from neurodegenerative diseases such as Alzheimer's or Parkinson's disease. Apparently, under these conditions, chaperones no longer do their job properly. Now, these molecules, these proteins, are called chaperones because they have similarity in terms of functional terms to the human chaperone. Their most important function, one could say, is to prevent improper interactions between not yet folded protein molecules that result in the formation of aggregates. In these aggregates, proteins cannot reach their functional state, and more importantly, a subset of proteins are able to form highly regular fibrillar type of aggregates, which are characteristic for these neurodegenerative diseases that I just mentioned. And in these diseases, we have beta strands a particular secondary structure element that runs perpendicular to the long axis of these fibrils. Now, the human chaperone helps people to find their way, just like the ambassadors we have here at our forum, and we are very grateful to them for helping us in so many cases. There are the chaperones that interact with many different partners. They just show them where the bus is. They can show this to me. They can show this to my colleague. And then we have the specific ambassadors. These are the specific chaperones that only take care of one particular person. In the case of molecular chaperones, there is promiscuity. They can interact with many different protein molecules, but the condition is that these protein molecules have to be in a not yet folded, as we say, non-native state, because during the folding reaction, polypeptide chains expose partially folded chains, expose hydrophobic amino acid residues to the solvent. And these are properties that are recognized by the chaperones. And by binding to these properties, to these sequences, chaperones prevent protein aggregation. And through binding and release cycles that are mediated oftentimes using ATP and other regulator proteins, they promote productive folding and suppress these aggregation reactions. So there is promiscuity on the one hand, but there is specificity for non-native conformational states. Prevention of aggregation is a major function, but more generally one could say that these chaperones solve landscape problems in the folding energy landscape that when left unattended can lead to the formation of these various types of protein aggregates, including, again, these fibrillar forms. Now, as I said before, during the folding process, you can accumulate various types of intermediate states. These are kinetically trapped states where the polypeptide chain has to overcome energy wells, energy minima, has to come out of energy minima to continue to fold towards the thermodynamically more stable native conformation, which is in most cases also the most compact state, starting out from a wide arrangement, a right ensemble of unfolded chains leading to this native conformation. And the chaperones help, they support the interconversion between these folding intermediates. They accelerate, they catalyze the movement of folding intermediates across the energy landscape and thereby speed up the overall folding reaction. Now, as I said, we have different types of molecular chaperones, and they act in ordered sequential pathways during the production of a protein chain on the ribosome, as it is shown here. These are the ribosomes. They read the information of the messenger RNA and build the amino acid chain that then emerges from the ribosome, and as it does, it immediately interacts with a first set of chaperones that we call nascent chain chaperones. These prevent early misfolding steps in the polypeptide chain when the protein is not yet able to fold because it doesn't have sufficient information. It is not long enough yet to specify the native three-dimensional state of the protein chain that has to ultimately be reached. These are molecules, for example, called HSP70 or trigger factor. These bind and release the chain elements that are otherwise prone to aggregation, and this can be sufficient for the folding of many proteins, but a subset then has to continue to use additional machinery, and these are particularly interesting large protein complexes. The chaperonin system, as we had described it in the mitochondria, it occurs in the bacterial cytosol. It occurs also in a somewhat different form in the cytosol of eukaryotic cells, in all of our own cells. And this machine provides a central space, a folding cage, as we say, in which a protein chain is transiently encapsulated and allowed to fold in isolation, thereby unable to interact non-productively with other protein chains that try to fold at the same time. So this is really a most fascinating and remarkable invention of nature to assist the protein folding process. Of course, the information for folding is in the amino acid chain, just as Anne Vincent said, but we need this machinery to implement this information in a functional protein. Let me now show you in a bit more detail how these particular machines, these chaperonins, work using the bacterial counterpart of the mitochondrial HSP60 as an example. These are ring-shaped molecules, cylindrical molecules consisting of 2 rings. Each ring has 7 subunits, and each ring encloses a central cavity. At the edge, these subunits expose themselves hydrophobic amino acid residues, sticky surfaces that recognize the sticky counterparts, the hydrophobic surfaces of an unfolded or partially folded polypeptide. These are the parts that are later, of course, buried when the protein is properly folded and can no longer be seen by the chaperone. Now, the protein chain is usually delivered by an upstream chaperone, such as HSP70, called DNAK in E. coli, which stabilizes the protein in a non-aggregated state and then lets it go, allowing its transfer into the central opening of the chaperonin. And now, when it has been bound, something really magic is happening. Namely, a second factor called GRO-ES, or HSP10 in the mitochondria, binds to the end of this cylinder, as shown here. It encapsulates the protein chain and displaces it into the central cavity, where it is now free to fold. At the same time, the environment of this cage that now forms changes from hydrophobic to hydrophilic and charged, so that now the polypeptide chain no longer interacts strongly with the chaperonin itself. It is encapsulated, it therefore unable to aggregate, but it is also free to fold. And it stays inside this chamber for a couple of seconds, timed by an ATP hydrolysis reaction that occurs on the chaperonin itself. After completion of the first round of ATP hydrolysis, ATP binds to the opposite ring, and this sends an allosteric signal. By that we mean across the ring plane to the side where the substrate protein was contained, and this allows the opening of this GRO-ES lid, and the protein product can emerge. If it is fully folded, it will not be recognized anymore by the chaperonin, but if the folding process was still incomplete, it will be recaptured very rapidly and goes through another round. Now, this mechanism, when we first described it, was highly controversial, I should say. It was a very challenging time for a couple of years to convince our colleagues that this is what really happened. And in retrospect, I have to say that this skepticism, of course, even though it was difficult at the time, was part of the normal scientific process. I'm saying this to you young scientists. So it forced us to do better and better experiments to test our model. Just as Mark Twain once said, continuous improvement is better than delayed perfection. But what would be the perfect experiment to demonstrate that the protein really folds inside this cage? Now, we also heard at the meeting that technical advances are very critical in allowing us to progress, to address questions in different ways, to get much deeper understanding of biological processes as in other disciplines. And in this case, what was of particular importance was the development of cryo-electron microscopy, a technique that allows us to solve structures of proteins, relatively high resolution. And this is exactly what we did for a protein that is now encapsulated inside the cage of the chaperonin. And we wanted to determine the structure of this end state of folding that such a protein reaches inside this machine. And this is shown here. This is 1/2 of the chaperonin cylinder with the GRO-ES lid, and encapsulated, you see here, the protein substrate. It's a protein of which we knew from earlier work that it is completely dependent on the chaperonin for its folding. and we can compare the structure of this protein with the crystal structure that is available for the folded METK protein that had been deposited in the database, and we find that it is essentially identical to the folded subunit of this protein. Once this protein emerges from the chaperonin, when the lid opens, these subunits assemble into a tetramer protein complex. That is then actually a spontaneous reaction, and this is the final active enzyme. So we now convince ourselves that protein folding up to this very end state really can occur in this system, and it does occur also inside the cell, as we have shown using a different version of cryo-electron microscopy more recently. Now, there are two aspects of this. One is that the aggregation of the polypeptide chain is prevented during the folding reaction, and at the same time, folding is accelerated, and we attribute this to an effect of entropic confinement, as we call this, whereby the conformational freedom of the polypeptide chain is restricted inside this narrow chamber, and thereby the polypeptide chain is allowed to find native interactions between distant parts of the polypeptide chain more rapidly than it would in free solution, and it avoids various kinetically trapped states along the trajectory to the folded protein. This way we can get rate accelerations of 10 to up to 100-fold compared to folding in free solution. Now let me shift gears. Obviously, understanding these fundamental functions, let us ask the question, why does protein misfolding and aggregation nevertheless occur, and why is this a problem? Why does it cause these terrible diseases, Parkinson's, Alzheimer's, that are not yet curable? Now, the protein folding chaperones are part of a much larger network of factors that we call the proteostasis network. And in this system, the functions of chaperones in de novo folding, in protein repair, in conformational remodeling of proteins are associated with the functions of protein degradation enzymes, most prominently the ubiquitin proteasome system, and lysosomal degradation, autophagy and lysosomal degradation. So the overarching goal is to avoid the accumulation of potentially dangerous misfolded states that can then give rise to these various forms of aggregates. We also understand, and you will hear more about this later, that cells can make more chaperones when needed under various conditions of cell stress, most prominently perhaps heat stress, but also other forms of environmental and endogenous stress, where proteins that have already folded begin to misfold and lose their folded conformations. But unfortunately, these capacities seem to decline as our cells age, and this is particularly relevant for cell types that cannot simply regenerate by cell division, such as neuronal cells in the brain and also muscle cells. And that's why this decline of chaperone function is associated with the manifestation, with the increase in the probability of the manifestation of these diseases. I mentioned them: Alzheimer's, Parkinson's, amyotrophic lateral sclerosis, Huntington's disease, various others. And they all have in common that we find aggregate deposits either within or outside of cells. And when we look at these deposits, they have these fibrillar structures that I already mentioned. Therefore, there must be some common aspects, not only with regard to the formation of these aggregates, but also with regard to the way in which they form cause cellular toxicity. Let's have a look at these fibrils in a bit more detail. These fibrils consist of stacked beta sheet strands, as I had shown schematically in the beginning. But this is not a real fibril, even though it has quite a bit of similarity to some of the more recently solved structures. This is actually the logo of the Hong Kong Laureate Forum, if you remember from the opening session. I found it quite intriguing that it looked so similar to one of these fibrils. But here is an actual fibril from cryo-EM analysis. The stacked beta strands, you can see there are various protofibrils that are arranged into the final structure, 7 to 20 nanometers in diameter. These are highly stable structures. If this were to be translated into the macroscopic world, a high-rise building could be kilometers high, not just 500 meters or 450 as the highest building in Hong Kong. So these are really very stable structures. We know this from their persistence lengths. Here is an image of how these fibrils accumulate in cells from a technique called cryo-electron tomography, developed by my colleague Wolfgang Baumeister at the Institute. It's a microscopic technique that allows you a look into an intact cell at macromolecular solution. So these are the molecules, the fibrils, as they exist in situ in either a HeLa cell or in a neuronal cells. And you can see that these spaghetti-like fibrils are surrounded by membrane structures. These are membranes of the endoplasmic reticulum. Again, you will be hearing a lot more about the endoplasmic reticulum in the next talk. These are sites where ribosomes associate these green particles and make proteins to be inserted into this membrane structure and secreted from the cell. But you can see that the membranes that are close to the aggregates, close to the fibers, are no longer containing these ribosomes because these membranes have changed their properties. And we think that these inclusions in various ways contribute to the disease process by displacing cellular structures. These inclusions are relatively large, and possibly also damaging these structures. Now, aggregate formation in disease follows a nucleation-dependent polymerization process, where we first have a primary nucleation event, usually the formation of a transient or incipient beta sheet structure, that then gives rise to soluble, relatively disordered oligomeric states, and finally to the ordered fibrillar structures. And there have been various ideas for why these aggregates and the process of their formation are toxic, such as the interference with membranes, but also the interference with other proteins and the sequestration of these proteins within the inclusions, an aspect that we have studied in considerable detail. And we have discovered that the aggregates also capture critical elements of the protein quality control machinery, of the proteostasis machinery itself, thereby setting in motion a vicious cycle that further reduces the ability of cells to fold new proteins or repair misfolded proteins. And then we observe more and more aggregation, and eventually the system then collapses. Now, we were interested in the beginning, of course, whether the normal chaperones that participate in folding, as I've shown you, can prevent this type of aggregation. And this is indeed the case. For example, the HSP70, one of the most ubiquitous molecular chaperones, acts very early on in this cascade by preventing the primary nucleation step. It keeps the aggregating disease protein in a more expanded state, and therefore prevents the formation of the nucleus. But other chaperones, as we found and others found as well, act at different stages of this aggregations cascade: primary nucleation, secondary nucleation, and fibril elongation. Now, what can we do to utilize our knowledge of these systems, of these protein quality control systems, to combat these diseases? Of course, we would like to find ways to pharmacologically correct the imbalance that exists in these disease states between the production of misfolded proteins and the available chaperone capacity. We would like to have a drug that augments this chaperone capacity. Now, we don't have such a drug yet that can be used in patients, but we do have a tool compound that at least shows that this could be possible in principle. And this is a cell culture experiment that I'm showing you here, done with the group of Erich Wanka in Berlin, where we use the protein huntingtin as a model, causing Huntington's disease. It forms these large aggregates. This is a fusion protein with the green fluorescent protein to make it visible. And when we treat cells with increasing concentration of this compound called galvanomycin, you can see that now the aggregation reaction is completely prevented. This is the DMSO control without the drug. So in principle, we can induce, and this is what this drug does, we can augment cellular chaperone capacity and bring it to a level where it is sufficient to prevent this type of pathological aggregation. So we are hopeful that in the future, our research and the work of many other groups will help in finding new strategies to delay the onset of these diseases and to increase healthy lifespan. And let me end with this beautiful painting by Lucas Cranach from 1546. That's the Ming Dynasty for the Chinese friends. It shows the Fountain of Youth. It's a world-famous painting. In the foreground of this fairy tale landscape, you can see this fountain and a pool in which old, frail women take a bath and thereby become rejuvenated. But there are many details in this painting that are really interesting and relevant to what I just told you. For example, here you see this person in the red robe, could be a doctor, who examines these elderly women as they are reaching the pool and finds out whether they are appropriate, whether they are good for rejuvenation. So this could be a kind of a quality control step, just like in the protein world. They then enter the pool, they become rejuvenated, they exit. They will be received by young gentlemen and dressed and then celebrate and party. So hopefully we will be able to find out what is the elixir in this pool. That's what we would like to understand and find correlates of this that help us to delay onset of neurodegenerative diseases. I've been lucky to work with a large group of wonderful people, students, postdocs, and also collaborators. But I want to stress in particular the contributions of Manajit Hayahardal, who is also here at the forum, who is an excellent scientist, and also my 24-hour collaborator, who has made a tremendous contribution to all aspects of our work. Thank you so much.
Thank you, Professor Hato, for that very, very intriguing talk. Please, can I invite you to join me in a Q&A session? I feel that, I can't agree more when Professor Hato shared how he utilized different technologies, right, to answer the same question. So, for example, he shared about using cryo-EM, right, which has just recently developed, to answer the same question that he did, back when when you got your award, and how you're able to verify the same conclusion, but now you're able to visualize this in a 3D manner, right? And I think it's important that I think science is intriguing in that we have all these technologies that come out, every year, and it's very important that we know how to utilize them to look at the same question at a different angle, right? And I think that's intriguing. So, please feel free to raise your hand if you have any questions for Professor Hato. So, okay, let's take that from the gentleman in blue.
Hi, great talk. My name is Alfred. I'm MD PhD student from Rockefeller. I think the difference between crystallization and cryo-EM is that in cryo-EM you really have a structural landscape in the native protein. I'm wondering, especially with recent technologies like time-resolved cryo-EM or these machine learning approaches to parse out the heterogeneity, a conformational heterogeneity, do you think we could actually, in what you showed in your cryo-EM structure, perhaps separate out the actual folding pathway and identify, because right now all we really have are MD simulations to.
That could be possible in principle, I hope, but it's certainly a lot more challenging than just looking at the final end state. That was already difficult enough, actually. But in cryo-EM, we can, of course, classify a wide range of conformational states and can potentially solve different intermediates along a pathway, or believe that we have seen a pathway along which we can then order those intermediates. That should perhaps be possible in the future, and it has been achieved at least in part for the eukaryotic version of the chaperonin that we call TRIG or CCT. This is the equivalent of the GRO-EL HSP60 in the cytosol of our cells, and it is required for the folding of such important molecules as actin and tubulin. And for both, we have a relatively good understanding of different steps along this folding pathway as it occurs inside the cavity of the chaperonin.
Thank you. I have a question from this gentleman on the right in a mask. We have a question over here up front. Yeah, thank you.
Hi, Professor Hato. Thank you so much for your sharing. I'm Billy Ng from the School of Pharmacy, CHK. So I have a question about one of your last few slides. You showed that there is a drug called GA, like a chemical pulp, which is really active in the nanomolar range. Could you share a bit more about how did you discover this molecule, and what is the mechanism of action of this molecule?
I did not discover this molecule, but I was at least involved in the early characterization of this molecule when I was still at Memorial Sloan Kettering Cancer Center in New York. We solved, together with the group of Nikola Pavletic, a crystallographer, we solved the structure of this molecule bound to its target, which itself is a molecular chaperone called HSP90. And HSP90 is a chaperone that participates in various signaling pathways. And one of the signaling pathways that is relevant here is the stress induction pathway, whereby a transcription factor is normally stabilized in an inactive state by HSP90, but under stress conditions is released from HSP90 and then induces the transcription of the various chaperone genes and allowing the cells to make more chaperones. And when we treat cells with this drug, we basically induce this pathway whereby HSF1 is released from HSP90. The problem why we cannot use this drug in people is that HSP90 has other important functions that we cannot inhibit. However, we could inhibit, and it would in fact be desirable to inhibit HSP90 in cancer cells, because HSP90 is required for the folding and activation of many proto-oncogenic kinases. And that is indeed a treatment, an option that is clinically investigated, but probably these inhibitors, if successful, would have to be combined with other drugs. There is apparently still substantial toxicity, and to my knowledge, this has not yet reached clinical application.
Perhaps just continuing on that, I was wondering, since you said that it might work for cancer, has there been any animal models that have been done for the use of that drug for cancer?
There are various animal models where you could reproduce the effect on the chaperone system, but certainly animal models to show that tumor growth could be inhibited. The reason why cancers, you have basically this interesting situation where in neurodegenerative diseases, you would want to have more chaperones, but in cancer cells, you want to have less chaperones. The reason is that cancer cells rely on mutant proteins, kinases, and other factors, growth factors. You need multiple mutations usually to accumulate in various components, and mutation makes a protein more strongly dependent on chaperone. So cancer cells are often chaperone addicted. So inhibiting chaperones could be a treatment for cancer cells as long as we could restrict it to the cancer and not hit the rest of the body.
Thank you. I have a question here on the gentleman in the white.
Yes, thank you very much. What was the main criticism that you had to overcome when introducing this new concept of that you presented?
Basically, they thought it was a crazy idea that a protein would fold within another protein, and the other team suggested that the folding reaction nevertheless occurs outside of the chaperonin, and that the main role of the chaperonin is to unfold the protein back to its starting state, to give it a new chance to explore the conformational space, so to remove misfolding and thereby bring it back to the upper part of the energy funnel. It is a valid idea, inspired by what had been learned about protein folding reactions from biophysical experiments in the test tube, but it didn't make sense to us, and apparently not to nature as well. So now this is well established, of course, and we did multiple experiments, as I said. It was almost a bit like a chess game, where we did something and then the other team did another experiment and tried to invalidate what we had done, or I shouldn't say invalidate, but to bring up counter evidence. Until then, the other team indeed changed their opinion. That was most satisfying, I must say.
So you won the chess game, right? So have you ever been discouraged to work on this area yourself?
Well, not to the extent that I would have stopped working on it, but it was challenging, and I must say that it was very good to have the support of Manergy during these days, during these months and years, in fact, because you can never be 100% sure that your model is correct. And it was good that we were challenged to do better experiments.
That's important. Other questions? We have one here on the left.
Well, actually, I have two questions. Is that fine?
Yes, please.
So the first one is that first of all, thank you for sharing. And I'm quite intrigued by the part that you say the idea of the caged ring is being controversial. I'm curious about, well, what exactly experimental assay have you used to, well, prove your idea is correct?
Okay, so there are two elements of the cage, as I said. One is that it prevents aggregation and at the same time allows the protein to fold. The other chaperones, like HSP70, they also bind misfolded or unfolded proteins, but they release the polypeptide substrate into the free solution. So it is given a chance to fold, but only for a short time. If it doesn't do it, it binds back and thereby avoids aggregation. Now, in the case of the cage, this aggregation prevention function is kind of obvious. What was less obvious and what came a lot later, the insight that perhaps an additional role and equally important was that the cage accelerates the folding of proteins that would otherwise only fold very slowly in free solution and would therefore undergo aggregation. The way we had to test this model was to perform single molecule experiments where we studied the kinetics of protein folding at the single molecule level. That means at very low concentrations, picomolar concentrations, where we could completely rule out aggregation. Because this would be the major criticism, of course. Again, we had, by the way, a similar debate. People said, well, of course, the folding reaction outside the cage is slower because you are observing transient aggregation. Maybe small aggregates form, but they are reversible and they are holding up the folding process. So therefore, we did the experiment at the single molecule level and could demonstrate that that was not the case. There was no aggregation, no transient aggregation, but the folding reaction was still substantially slower. Now, why the folding reaction exactly is accelerated inside the cage, I would have to say is not 100% understood. A simple model is the idea of the conformational restriction, but there are also other effects of the physical environment of this cage. For example, it has a strong negative net charge, and the majority of the substrate proteins that utilize this system also have a negative net charge on the surface. So there could be some repulsion effects that also play a role in the compaction that the molecule experiences when it is encapsulated inside this cage. What we did, perhaps the best experiment to demonstrate this principle, and this was really the idea of an excellent Indian postdoc who worked with us. We made mutations in a protein that had otherwise a robust in vitro folding pathway. We made mutations that disabled its folding reaction and made it highly dependent on the chaperonin for folding. Once it was folded, this protein is quite stable, but to reach the folded state, it now required the chaperone. This, by the way, is an important principle whereby these chaperones probably and very likely supported the evolution of new protein structures, which of course occurs by the acquisition of mutations in a pre-existing protein. And those mutations generally make the folding reaction less efficient. Now, we observed that this protein was now chaperone dependent. We observed that there was a tremendous acceleration of folding for the encapsulated state over the soluble state, even at single molecule level. And when we now inserted disulfide bonds into this protein chain to stabilize it in a more compact state, we could basically mimic the effect of the chaperonin cage. So this is really perhaps our best experiment that suggests up to this point that there is really a compaction effect. More recently, we have developed a system that allows us now to unfold the protein while it is inside the cage. The cage is very highly temperature stable, and we made it more stable. And you can then study its refolding, and you can study the stability, the thermodynamic stability of the protein while it is encapsulated. And we can see that the cage substantially stabilizes the native state of a protein. Again, another discussion that will probably ensue from this. But it's a very interesting finding that supports this idea that the cage profoundly modulates the energy folding landscape.
Thank you. It's really fascinating. Another question I would like to ask is about the neurodegenerative part you mentioned about the chevrons. I come across some recent papers talking about the liquid-liquid phase separation of small molecules in cells, which have a really similar idea, I think, with what you mentioned, that oligomers form aggregations in cells.
Yeah.
What I'm thinking is that would it be possible for what you suggested, the chevron models, increasing the chevron's concentration or something like that, can it prevent the LSP to take place, or is it actually similar things? I would like to hear about your thoughts.
That's a very interesting point that you're bringing up. There is a more recent development in biology in understanding that there are subsets of proteins in the cell that can undergo this phenomenon of condensate formation, phase separation from into a liquid-like state, and this is also related to the formation of these pathological aggregates, whereby it has been demonstrated that some of these disease proteins, such as alpha-synuclein in Parkinson's or Huntington in Huntington's disease, they first may form these types of condensates, and within the condensate then some hardening of the condensate occurs, some formation of more stable gel-like aggregates finally ending up in these fibrillar states can occur. And we and others have found that chaperones can insert themselves into those condensates and within the condensate interfere then with the formation of these types of pathological aggregates. So condensate formation was probably also very important early on in the evolution of the first life forms, because you're probably familiar with the idea that the cellular environment is a very highly concentrated macromolecular solution. It would have the consistency of a viscous honey. It's almost impossible to create in the test tube this type of concentration. So there must have been forces to bring molecules, not only protein molecules, but other molecules together, probably through this phenomenon of phase separation.
Thank you.
All right, so in the interest of time, we will have to wrap the session up, but if there are questions, please leave them to the panel discussion at the very end, okay? So thank you very much, Professor Hato. So our second speaker for this next session is Professor Katsutoshi Mori, apologies. Of course, Professor Mori is a professor of biophysics in the Graduate School of Science at Kyoto University, and of course the 2014 Shaw Laureate in Life Science and Medicine for his discovery of unfolded protein response of the endoplasmic reticulum. And so may I please invite Professor Mori onto the stage, please. Thank you.
Thank you, Stephanie, all kind introduction. And it's a great pleasure and honor to be here to talk about our analysis of unfolded protein response. So Dr. Hato told us how chaperone helps protein folding. I talk about the regulation of chaperone expression levels made by this unfolded protein response. Chaperone is not a special scientific word. This is a famous picture by Lenoir. She is a chaperone, a person in the past who went with a young unmarried woman, she is here, to social event in order to make sure that the woman, young woman, behaved properly. That means molecular chaperone helps protein to behave properly. That's the original meeting. Now, how many cells do we have? Do you know? 60 trillion cells. Some cells, 37 trillion cells we have. Then we have many, many organs. These organs communicate to maintain health. For example, you had lunch a couple of hours ago. Immediately after taking meals, your blood glucose levels increased. But this increased glucose decreased rapidly. This is very important because blood glucose levels kept high for a long period, you develop diabetes. This glucose level must be decreased. You know what's the mechanism, Oscar? Pancreatic beta cells, upon meal, upon high blood glucose level, pancreatic beta cells secrete hormone called insulin. On the other side, liver cells, a muscle cell, express insulin receptor on the surface. Then insulin binds to its receptor. Then this liver cell, mast cell, take this blood glucose into their cells. That's why blood glucose level return to the normal level immediately. Okay, this is very important. So organs have intercellular communications. We say that this is a key and keyhole relationships. Insulin must have this structure. Insulin receptor must have this structure for specific binding. The question comes, where these proteins are folded? Already Dr. Hato mentioned that compartment surrounded, intracellular compartment surrounded by membranes are called organelles. When you looked at a cell by microscope, it's simple. You see nucleus and mitochondria, maybe Golgi apparatus. Nucleus contains DNA, genetic material. Mitochondria produce ATP, energy. Is these enough for life? It shouldn't be. These three scientists, Rockefeller University in the United States, utilize electron microscope to watch the cell Then they found cell contains many, many organelles. Lysosome, peroxisome, and endoplasmic reticulum, ER, which I work on more than 30 years. Then, but I said, we cannot see ER using microscope, but Dr. Shimomura discovered very interesting, important protein called GFP, green fluorescent protein. Upon laser exposure, GFP turned green. So we can see ER, this is ER structure. We see using a normal fluorescent microscope. We don't need to use electron microscope. That changed cell biology field. So he got Nobel Prize in 2008. This is what a high school student will learn in a cell biology class after entering university. These three scientists, or Dr. Parade, discovered using electron microscope and the ribosome, and he found that it is a site of protein synthesis. Also, these scientists hormone is secreted from the cell. They are interested in where hormone is synthesized, how these hormones go out from the cell. Then they discovered the hormone, like insulin, is not synthesized in the free ribosome, but synthesized at the ribosome attached to the ER. Then hormone goes through the ER, then goes through 3 layers of Golgi apparatus to be secreted. Then, due to this great discovery, they got a Nobel Prize in 1974. Second generation scientists, represented by Dr. Brober, asked why hormone is synthesized, ribosome attached to the ER. Then he discovered the signal on the hormone that target a hormone messenger RNA to this ribosome. Then he got Nobel Prize in 1999. Third generation scientists asked how proteins moved from ER to the Golgi to the cells outside the cell. Protein does not swim in the cytoplasm. They get on the boat to be transported from ER to Golgi. I talk about this process in more detail later in my talk. Then they got Nobel Prize in 2013. We are in a fourth generation. Protein folding is important for the protein to be transported from the ER to the Golgi. Because insulin, secretory protein like insulin, or transmembrane protein like insulin receptor are folded in the ER. So ER contains a number of molecular chaperones quite abundantly that assist folding of newly synthesized protein. The only correctly folded molecules are allowed to move along to the secretory pathway, move to the Golgi, and move to the cell surface or out of the cell. However, Dr. Hartle showed that more than 90% of newly synthesized protein are folded properly and reach final destination. But cell makes large number of proteins. So some are still unfolded or misfolded even after assistance of molecular chaperones. These are problematic because, as Dr. Hartle said, so they are sent back to the cytosol to be degraded. So ER has 2 systems, chaperone-mediated folding system and degradation system. By these 2 mechanisms, they check quality of synthesized proteins. However, under a variety of physiological and pathological conditions we correctly call ER stress, this quality control system is compromised, resulting in the accumulation of unfolded misfolded protein in the ER. Because, as I mentioned, only correctly folded molecules are allowed to move along the secretory pathway. This prolongation ER stress, cells suffer from the shortage of protein at their destinations. So ER stress is detrimental to the cell. How the cell can cope with this ER stress? The cell activates unfold protein response. What's the best way to deal with this situation? Okay. Cell consider we don't have enough fun. That's why problem occurs. So to cope with this stress, cell increase chaperones to make more productive folding, more protein folding, and then folded protein, allow folded protein to the destinations. And also increase these destroyers. Then due to this more folding, more degradation, cell can maintain the homeostasis. So, and the ER is a factory, and under ER stress conditions, the cell induce chaperones and destroyers. So we wanted to know the mechanisms. This is a system, but no molecule was identified here. Because when I was a freshman at Kyoto University, molecular biology showed up. That fascinated me. I wanted to learn molecular biology, but molecular biology was a really advanced science. I could not get closer to it until age 30. I was a biochemist until age 30, but I thought I cannot, under this condition, I cannot do with molecular biology. I decided to go to United States, learn molecular biology. Then I went to Texas. Then I met this system for protein response. I was quite fortunate. There is a system exists, but no molecule identified at that time. So I could start from the beginning of this analysis. Then this system was originally identified in the mammalian system. The mammalian system is too complicated. So both decided to work on yeast unicellular organisms. Probably UPS system is more simpler, more easy to analyze. That was a great decision. So yeast cells form colonies on this agar plate. Single cell divide, make this one colonies. Okay? Then usually colony is white. I did gene engineering. When chaperones are induced, I made this cell turned blue. Then if we add the DNA mutagen, okay, then mutagenize DNA randomly. If this mutagenesis occurs in the signaling molecule, the cell cannot turn blue. Cell remained white. Then look for the gene involved in this UPR system, signaling. Because this random mutagens, very rare to hit the target gene. So need to screen many genes. Because success of this screening changed my life. I may be able to continue research. So I decided to work hard as much as possible. Then I screened 100,000 colonies. It took half years. Then I got 3 white colonies. Then next was what kind of gene is mutated in this colony. Then I discovered one gene. All 3 colonies has a mutation in one gene. The sequencing of this gene showed that it's a sensor in the ER. This sensor recognizes ER stress accumulation of unfolded proteins. Then next, sensor is not enough. Sensor can recognize ER stress, but cannot induce chaperones. Chaperone is induced after years, but this induced chaperone is not walk around under normal situation. You can consider that these chaperones are sleeping in the nucleus. So if sensor recognize this emergency, let's send emergency signal to the nucleus to say, wake up chaperone, go to ER to help this situation. To hold this one, you need a wake up call. That means drummer hits drum to make wake up call. this drama is called transcription factor, activate transcription in the molecular biology. Then this one is increase. So I discovered this drum first in 1992. Then I and Peter Walter at UCSF discovered this sensor in 1993. Both of us, then I went back to Kyoto, and me and Kyoto and Peter Walter at UCS discovered drummer. So there's a severe competition, but because of the severe competition between two laboratories moves this field dramatically forward. Then Peter and I shared 5 major international awards, including Shaw Prize. So next is, how about mammals? Similar system must exist. Then I recruited a young talented scientist, Hidero Yoshida, and asked him to find a drum. And he was talented enough to identify drums in 1998. Then we discovered the sensor in 1999 and discovered the drummer in 2001. So we sort of, also other scientists get involved in this mammalian UPR system. By the end of 2001, we unraveled most of the basic mechanisms of the mammalian UPR. So this is a tree called evolutionary tree or phylogenetic tree, how various organisms are evolved from ancestral prokaryotic cells. We are here now. Then first, prokaryote cell appeared. It doesn't have any organelle. DNA, protein, RNA exists in a kind of mixed situation. But next, budding yeast shows up. This one makes my favorites here. And these yeast cells much bigger than prokaryote. Because its volume is more than 1,000-fold. Then these eukaryotes move rigorously. That cause DNA damage. So to avoid DNA damage during movement, they created a nucleus to encapsulate DNA. At that time, ER was formed. Then ER has one sensor called IRE1. Next, multicellular organisms showed up, like C. elegans, called worm cells. It consists of 1,000 cells, and then various organs. So in this worm, we found 2 sensors. IRE1 conserved from yeast to C. elegans, and second sensor called PERC. Under ER stress, protein folding is compromised. Protein folding doesn't work well. If a cell keep synthesizing protein and keep delivering protein in the ER, we have more ER stress. That's the problem. So this sensor stops translation when unfolded protein accumulate. Then during that time, induce chaperones by IRE1 to maintain the homeostasis. In mammals, we found that they have 3 sensors. Show that what's the role of these 3 sensors in the maintenance of our body. We create knockout mice. When IRE1 pathway is knockout, mouse cannot be born because they have a poor development of liver. Liver is a major source of protein circulating the plasma. So maintenance of large amount of protein is important, require this IRE1 system. PARC knockout mice developed by diabetes. They cannot maintain the synthesis of large amount of insulin. they suffer from insulin cause diabetes. Knockout of ATF6 cause also embryonic lethality. Mouse without ATF6 cannot be born, die very early stages. Notochord has a problem. Notochords are axis before the bone is formed. A very important one at the early development stage. So really these ER sensors are important for our health. Then yeast has only IR1. This IR1 takes care of the chaperone levels. Then C. elegans, also IR1 takes care of the chaperones. But knockout mouse examination showed that in mice, 8-6 take care of chaperone, not IR1. So there must be a switch from IR1 to 8-6. We hypothesize that this change occurred with the development of backbone. Then we analyze small fish called medaka fish. Probably you have a different name in Chinese, but probably you know this fish is associated with rice making. You can find this one, water rice field. So then we found that it's true. 8-6 take charge responsible for the chaperone expression levels. We're still working on why major regulator of chaperone was needed to be changed from IR1 to 8-6 with development of backbone. It's still a question. Also, during the, evolution, we see another changes. Number of this type of sensors is 2 in non-vertebrate, but 5 in vertebrate. So why we need so many sensors? If ER stress simply means accumulation of unfolded or misfolded proteins. 8-6 is ubiquitously called expressing all cells we have, called ubiquitously expressed. Also, this lumen is also expressed ubiquitously. But the CREB-H is expressed only liver and gut, as in mice. Oasis is expressed only on the bone tissues. So these sensors might cope with local ERS, not general ERS. So we make all of knockout medaka fish and analyze. Interesting results came with BBF2H7. Key molecule was that one was collagen. You know collagen, most abundant protein in our body. 20% or 30% of protein we have is collagen. That's collagen is ladies are like collagen because good for the maintain beautiness. But this is a protein. Okay, the protein synthesized the ER bound ribosome. And they fold it. But collagen supports cell from outside called extracellular matrix protein. So collagen is folded in ER, need to be secreted to the outside of the cell. Chaperone helps folding this protein. So 8-6 supply enough amount of chaperone to assist folding of collagen molecules. Then this protein needed to be transported. In this case, this one get on boat to be secreted. But sometimes we make long proteins, big proteins. Usually this boat has a diameter of 60 to 80 nanometers. But sometimes we made a protein of more than 300 nanometers. It cannot be accommodated into the normal boat. Then, in this case, this sensor, BBF2H7, recognize the appearance of long molecule, then make a tankable ship to accommodate this long chain molecule. Then, so this one, due to this enlargement of this vesicle, this long chain collagen is secreted to outside cell to maintain health of cell. So we think that UPR provides backstage support for various biological processes. Insulin and insulin receptor is the main actors. That interaction maintain blood glucose level low. So they're the main actor, but they need support from UPR for folding. Also, in the case of type 2 collagen synthesis, they need support from UPR for enlargement of vesicles for the transport. Then its failure leads to developmental and progression of various disease, like obesity, insulin resistance, like many cells. So we and many scientists in the field, we are tackling the problem, how to treat these diseases. Thank you very much for your attention.
Thank you, Professor Mori, for that very nice talk. Can you join me? Yeah, please, in the stage for the Q&A. So again, like last time, please raise your hand if you have a question for Professor Mori. Over there, this lady on the left.
Thank you so much, Professor Morley, for a very interesting talk. It was actually pretty hilarious at sometimes. I really enjoyed it. I have a question about the evolutionary history of ATF6 and why suddenly we evolved essentially to stop using IRE1 and to use ATF6. Was ATF6 a result of like a gene duplication event that somehow ended up being better at the job than IRE1? And in systems where currently ATF6 is the main actor, then what is IRE1 still doing? Thank you.
I didn't have a time to mention that all metazoan cells contain ATF6, but in non-vertebrate, it contains ATF as a single gene, but no role in UPR. IRE1 is a major regulator of chaperone levels. Or ATF has doing nothing. But after formation vertebrate backbone, vertebrate contains two ATF6 genes. We call H alpha and beta. Then it's gained function in the UPR. The IRE1 system is also transcription induction. Finally, transcription factor is activated. But regulation is complicated. Messenger RNA encoding this transcription factor must be spliced. RNA must become shortened, and spliced RNA needs to be translated. So it takes time. Splicing, translation, 2 steps. In the case of ATF, it exists as a precursor. Then just cleavage activate ATF6. So cleavage pre-existing protein quite rapid, much faster. It's still under investigation. That answer?
I actually have a question, because you showed in your last slide that UPR, of course, is implicated in a lot of diseases, including cancer. And UPR is a pro-oncogenic mechanism, right? Can you enlighten us on anything about, targeting UPR or its related network for cancer, like in translational aspect? Thanks.
Most in the clinical aspect, chaperone is a protein. It's difficult to deliver to the cell. But scientists found a small molecule that can stabilize protein folding status. That's called chemical chaperones. Okay? Not a protein chaperone. Small molecule chaperone is called chemical chaperone. That assists folding status. In Europe, chemical chaperones are used to treat liver diseases. Some review said it's used with safety and efficacy. There are also some chemical chaperones that shown last year or 2 years ago, shown to delay the progression of ALS symptoms. That chemical chaperone were approved in the USA and Canada for treatment of ALS patients. But dose is gram. 3 gram and 1 gram, 2 combination of chemical chaperones. You must eat, like a patient must eat like a cookies every day. So it's a better treatment is necessary. So that's most close to the in clinical state. There also UPR is important for cancer progression. At the beginning, cancer suffer from hypoxia and poor nutrition conditions. To activate UPR for survival. Some scientists discovered IR1 inhibitor. That in the phase 2. So some approach is being made.
Oh, that's encouraging. Very. Questions from that lady? Yes.
Hi, Professor Mori. Thank you for your talk. I was wondering if you could elaborate a little bit more about what is actually recognized in a misfolded protein, like considering if you have like essentially one way to fold it correctly and maybe a billion ways to fold it incorrectly, and potentially very large proteins with only a small part that is misfolded. How is that actually recognized?
Yeah, yes, it's critical question, but in debate for 30 years. So 2 models exist. And IR1 is, I didn't show it, also this lecture to the ordinary people. IR1 is like a protein kinase activated by dimerization. Then chaperone usually bind to IR1 to prevent dimerization. When unfold protein accumulate, chaperone move to the unfold protein, then allow dimerization of IR1. That activates signaling. So chaperone mediated sensing is one model, but Peter said that IR1 itself can bind to misfold protein. So direct binding of misfold protein to IR1 activate UPR signaling. So it's in debate, no any conclusion. But we are doing experiment distinguish 2 process. Our result is close to Peter Walter's story, the direct activation model. Okay? The chaperone recognizes unfold protein and misfold storage, as Dr. Hato said, misfold storage. Really, chaperone does not recognize the entire structure. If hydrophobic storage is open on the surface, chaperone binds. Chaperone cannot discriminate other part of the protein. Just problem is exposure of hydrophobic switch or no. Direct binding case, some specificity exists. In one tissue, like a liver, maybe has misfold protein that directly activate IR1. Well, in that PARC is also activated dimerization. In the pancreas beta cell, misfold insulin may preferentially bind to PARC to activate signaling. That's another possibility.
Thank you very much, Professor Mori and Professor Ma. Thank you very much. Please remain on stage as we now move to the panel discussion. Please take your seats on the right, and we'd like to invite Professor Hartle to join our guests on stage again. Please welcome our scientists with a big round of applause. Now, in this section, we'll have a chance to further discuss and exchange on the presentation materials by our speakers and also any related topics that are related to their research experience. Not to mention, our panel discussion does allow some questions from the floor and also from our online app, the HKLF app, mobile app. And now, without further ado, over to you, Professor Ma.
Okay, thank you, Tasha. So this will be a discussion, a panel discussion, I guess, which is essentially continuing on our Q&A question, and then also, so if any of you here that have any questions for both of our laureates, please, again, feel free to raise your hand here. We have the same lady that was going to ask Professor Hato a question. Please.
Thank you so much. It's great and really inspiring. I'm Jennifer, from a postdoc fellow from Canadian Institute for Theoretical Astrophysics, and then an Institute of University of Toronto. So I'm from very different background. I'm trained as a physicist. But looking at the protein folding is really fascinating. And it kind of drew me to think about like magnetic field in space. We talk about also the folding, twisting, how it links, how it tied to make a knot. And I was thinking, do we see a protein, not just folding, but actually make a knot? And for a knotted protein, is it something that is disastrous or is it something good? So I'm kind of thinking about like the broad picture and get some inspiration, wisdom from you. Thank you.
Well, there are actually knotted proteins where the protein chain moves through a loop of the protein chain, of another part of the protein chain, forming what would appear like a knot-like structure. So, and in fact, it has been seen that some of these knotted proteins utilize the chaperonin for folding. The exact reason is not understood, but I would perhaps suggest that, again, the enhancement of compaction inside this space might be utilized for forming the knot.
Oh, thank you so much. It's really
It's something that
great,
stabilizes a protein structure also.
So it's actually to enhance stability?
Presumably, yeah.
Okay, cool. And is it good or is it bad? It sounds like
if this is the native structure of the protein in which it is functionally active, the protein has to adopt this structure in order to perform its role.
Awesome.
So you would say it's good.
Yes, awesome. Thank you.
I actually see that we have a younger group of audience who are from secondary school students, those that are in the uniform, I believe. So it's okay, don't worry. I'm not asking you to ask a question. They're all nervous. So I guess, but when I go, but I'm sure, one of the questions that they may have in mind is how the two professors actually started in doing what you do now. Is it because you joined, so happened to join a lab that was just working in that area, and then you just sort of followed what your supervisor was, asked you to do, or, was it something that intrigued you and that you actually, how did you start, in your field? How do you start it?
Yeah, as I mentioned, that I really wanted to learn molecular biology. Then, so I took a chance, and then I decided to go to United States. I didn't care about the project, just want to learn molecular biology. First choice was learn molecular biology. Second is a project. I wanted to do neuroscience, but I sent 3 airmail, not email at that time, 30 years ago. So I sent 3 airmails to the professor in neuroscience. They couldn't accept me. So then I sent an airmail to boss in Texas. He accepted me. They found that this UPR system really fascinating. At that time, many researchers doing the signaling from outside the molecule. When ligand bind to receptor at the surface, then how signal transmitted into the cell, the PDGF or EGF. They're very competitive field. But UPL concerns the intracellular signaling from ER to the nucleus. That's something new, I thought, that there might be a very interesting molecule or system there. Then it is indeed it is a case as expected. Very unusual splicing where proteolase is utilized to activate signaling. So since then, I fascinated UPL, then kept working on more than 30 years.
So Professor Mori, I guess it was driven by you wanted to learn molecular biology, and then it so happened you went to a lab that worked on UPRs, and then you found a field that you found, that you thought was a gap, knowledge gap, and you wanted to work on. How about Professor Hartel?
Yeah, well, it's so that I was already as a little boy, I would say, or in my early teens at least, became very interested in biology and chemistry. My grandfather was a hobby microscopist and let me look through the microscope to see cells, and he studied fungi actually. So, and later I collected insects, butterflies and beetles. My father gave me a little chemistry lab. We did some simple experiments together. He told me that he was passionate about doing such experiments when he was a boy, except that his father always took the chemistry set away from him and sunk it in the Rhine River. And that must have happened at least twice or so. And my father, I remember, full of pride, said to me, but 2 or 3 weeks later, I already had everything again. And yeah, high school was very important for me. I had wonderful teachers in high school, biology and chemistry. And somehow it became clear to me that I would like to be a biochemist, but I did study medicine. And that was actually a good choice because at the time I could spend a lot of my time in the biochemistry lab early on and started to do my own experiments, much earlier than a chemistry or biology student. And I have found again a wonderful mentor who I have been very good friends with till today. He was at the time setting up his own lab, and I was a kind of apprentice with him. And my doctoral thesis was graded by Walter Neupert, who became then my next mentor, where we did the work on mitochondrial protein import. He was already a famous scientist at that time. One step after the other, just completely not really predictable or planned. We went to the US. Before I met my wife, managed it. We then went to the US. We went back to Munich. We went again to the US. And then finally to Munich again, to the Max Planck Institute.
Interesting. Must be a very proud grandfather.
Well, he has passed away a long time ago.
Questions from the audience? Please. Yes, lady at the back.
Thank you very much for this interesting session. I'm Chitra Joshi from University of Oxford. I have a question regarding like a future perspective of this mechanism. So we now know how the protein, the misfolded proteins are identified and degraded. Do you think in future we can develop or identify how the misfolded protein can be refolded? Like, can we correct the folding? And if so, are there any evidences where people are working on this, or what are your opinions? Thank you.
Currently, most successful example of this research is molecule-targeted therapy. The antibody was used to cure diseases. Like Dr. Ferrara developed anti-VEGF or vesicular endothelial cell growth factor or anti-VEGF receptor that stopped progression of cancer or progression of some eye diseases. many examples of molecule-targeted therapy. But in that case, antibody targets ligands or cell surface receptor. So injection of antibody into the body easily have access to the targets, the ligands or cell surface receptor. But our UPS system works intracellularly. It's very difficult to deliver antibody inside cell, block activation process. Then people use small molecule. But small molecule does not always work. It's some very difficult non-druggable target exists. So I struggled for a while how we can use this UPR system to cure diseases. But Dr. Calico changed the world. She developed mRNA technology. We injected spike protein of COVID-19 virus. That works very well. Yeah? So I thought that if we can deliver target molecule by injecting mRNA, then increase or activate signaling, well, even block signaling. So in next decade, maybe we can develop intracellular molecule targeted therapy. That's my dream.
That's very interesting. Thank you. I can pass to the front. Yes.
All right. So my research is something very different in mathematics. And in mathematics, a lot of people think that maths is about just, I don't know, maybe they think mathematicians just add to big numbers every day and just do calculations. And I wish more of the world knew that maths was about ideas and problem solving and things like that. So my question is there a medical and life science version of that, a misconception that people have about the discipline, or something that you wish people knew about the disciplines? This is a much broader question, I guess.
You mean that there are still major unresolved problems in our discipline? Is that what you're referring to?
I mean, is there, I can imagine that there's some children who think that, oh, I like mixing things in the kitchen, so I should be a chemist. I guess that's not what chemists do, right? So is there something that misconceptions that people have about what you do in general? What is the nature
but basically,
of
we are playing around in the lab. And we have ideas. We are simply curious to understand a particular reaction in our case, in cells. We are working mostly at the cellular model, or we even work with further reduced models where we isolate the proteins that I described and study their functions in the test tube with more and more sophisticated techniques. But basically, we are just curious how they work. And we hope to discover something. Discovery means it's new. It's not been seen, or at least not been understood in that way by anyone else before. And that feeling when you make such a discovery, even a small one, is an amazing, exciting feeling. And I guess that's what's common in all the sciences and disciplines that we heard about at the forum in different ways. Physicists, astrophysicists, life scientists, mathematicians, they make discoveries, and that's what keeps them going. I think Michael Welch pointed this out in his talk very well, and also others. This excitement, if you experience it as a young person, as a young scientist, you become addicted to it. This is what you really want to feel. And in the beginning, of course, it's often very challenging. Most of our ideas don't work out. Most of the experiments don't work out in the way at least we hope they might. But eventually something works, which gives you then hopefully the confidence that you can do it. But if you then somewhat successful, you will have coworkers, and they have their failures, but perhaps 1 out of 5 or 1 out of 10 makes a discovery. And that makes the whole process a lot easier, because failure is no longer such a major problem. And you are interacting with young people, which keeps us young, I think, or at least that's my hope. And so it's a different level of excitement again that makes this such a wonderful, privileged profession. And the whole idea of this forum, of course, is for us to convey this to you, young scientists, perhaps to convince some of you to seek a profession in science.
Very well said. I think, Professor Mori, you have more comments?
During we are a student and postdoc, we have to work so hard. But once you become PI, you can determine what you want to do or what to do next in your life. So that's fascinated me. No? But if you go to the company, it's one of a small gear. Then company told you, you go to this one, this one, this one. Then your hope is not important. Company decide what you do. So that become a PI. Then you like it.
Then you are in the driver's seat.
Thank you.
So stay in academics, right? Your question right behind, yeah. Yes.
Thank you, Professor, for your sharing. I'm Wyman, so I want to ask a question out of my curiosity. So for cancer, it usually takes up to more than one mutation in the cancer suppression gene to cause a cancer. But so I want to ask if there's a mutation in the chaperone gene, is it sufficient to cause any protein folding diseases? Thank you.
There are various cases where mutations in chaperone genes can contribute at least to a disease or dispose. These are usually not the major chaperones that are mutated, but rather the regulatory factors of the chaperone system that we call co-chaperones. For example, in the case of HSP70, which is a major chaperone machine in all of our cells, we have up to 50 or so different co-chaperones that regulate the function of HSP70 or recruit HSP70 to particular sites in the cell, to particular protein substrates. And defects in such chaperones can produce a much more restricted loss of function that then can give rise to a disease, but it's not a lethal condition.
Thank you.
Continuing on to the gentleman on the left. Yeah. Thank you.
I was wondering, due to an earlier statement, do you still work in the lab yourself nowadays?
Who is asking? I cannot. Okay. The answer is no.
No, because I thought because of an earlier statement, and then maybe you say become a PI. Of course, there are some downsides to becoming a PI. How do you prevent these downsides, like trying to get as much funding, which maybe in your position is less hard as an established investigator? How do you prevent these downsides and really try to focus on the science itself?
Yeah, me neither. I wanted to do experiment, and when I became an associate professor of Kyoto University, we have a limited space for experiment. I didn't get enough space for my experiment, so that's why I stopped experiment at age 40. Then after that, I write a grant, write a paper, and that's my main work. Then recruit good student. Then go to meeting or lecture as much as possible. Then get good student is most important. Fortunately, I can obtain good student. When we want to work on mice, guy working with mice came to my lab, and that kind of thing. So important was interaction with young scientists and make them interested in my research. Okay.
Would it be fair to say that at different stages, you do different things, right? I mean, of course, as a PhD, as a postdoc, you're focused in the lab doing the hands-on work. But then when you move to become a faculty, of course, then you train the next generation. You're still doing science. It's just that you're talking to them, and communicating with them, and then letting them do it. But you're still there to do science and guide them. I guess that would be fair. But of course, there's also a lot of other administration duties and teaching duties that comes along.
But the challenges that you referred to, with regard to grant writing and things like that, I mean, they remain. Last year, for example, I applied for a new ERC grant from the European Research Council. These are the most competitive but very large grants, and I wasn't really sure that I would get it. I mean, it was putting me back to giving me the feeling at a much earlier stage of my career, where you are suddenly again examined by your peers. You have to go through an interview. You have to answer questions. Why do you want to do this experiment? Why are you not doing this or that? And of course, I wouldn't tell you this if it wasn't successful. But it's still the same. So not maybe to the same extent as when you start out, but when you start out as an assistant professor, you usually get a nice startup package that should allow you to do research for at least 2 or 3 years, and hopefully during that time you will be able to produce a first piece of work. And as Michael has pointed out in his talk, it's not necessary to be one of the top journals where you publish your first work. Just put something out to show that you are alive.
My school has an undergraduate program, so I had a heavy teaching duty. I had to teach 50 times a year to undergraduate students or cellular biology, molecular biology. But I thought my obligation to hold the next generation. I worked hard to hold that teaching, so that decreased my research time. But I think I have to do it for the next generation. I did it. I retire from my school next March. I have no teaching duty, so I'll be able to do more research from next April, I hope.
I believe there was a question.
Hello. So piggybacking off of the recent question on working in the lab, I was wondering, since my favorite wet lab experiment is the Western blot, if you have a favorite wet lab experiment, and if so, if you have, for example, 1 week free from all your teaching duties, free from everything, grant writing and everything, and you had the chance to do an experiment in the lab again, what would you do? Thank you.
Tough question.
Maybe I would repeat one of our earlier experiments that brought us so much happiness, a in vitro reconstitution of the chaperone function, in which we combined 2 sequential chaperone systems, whereby the first one stabilizes the protein against aggregation, and the second one then folds it to its native state. This experiment we did in 1992 for the first time, and we had to purify all these proteins, recombinantly express and purify, which was, of course, a lot more work at that time. And the PhD student who did this work, who is now himself a director at the Max Planck Institute, Thomas Langer, When he did the experiment for the first time, it immediately worked. It was almost unbelievable. But unfortunately, it stopped working then because some of the buffer solutions went off, and we had to first figure out what was wrong, so we remake all these reagents. But since then, I have to say it has always worked, and many of the people who went through my lab have done this experiment again to familiarize with themselves with the system. And I think that's probably the experiment I could perhaps still do if the technicians prepare all the protein solutions for me.
I like to acquire new technology, like a GFP expression. It's dramatically change cell biology field. Then, when genome editing was emerged, we quickly get access to that techniques. That changed a lot because before genome editing, if you want to make knockout cell, you have to make knockout mice, then isolated fibroblasts, knockout fibroblasts. It's laborious, and if you have 2 genes, 3 genes, you have to make triple knockout mice. That is expensive and then laborious, take years to get knockout cells. But in genome editing, we can get any gene knockout in a normally culture cell. Right? Then we had introduced in the field of we call ER-associated degradation, how misfolded proteins are recognized and delivered to the cytosol to be degraded by ubiquitin proteasome system. Previously, people use there are 3 genes, people just use overexpression to see effect. But I thought it's not a good way, but we had no way to get knockout cell before. But after genome editing, we could make any single knockout, double knockout, triple knockout cell. That changed the field dramatically. Previous guess was not right. We could get new discoveries by obtaining genome editing techniques. So RNA-seq, many new techniques come every year. So just keep going forward is our policy.
So thank you very much. This brings our panel discussion to an end. That was a very intriguing dialogue. And so please give our hands up for Professor Mori and Professor Hato. Thank you.
Thank you. A big round of applause for them. Thank you so much for sharing with us your valuable experience and also the journey along the way with some interesting stories. Thank you very much. Please take your seats with our audience. Thank you. Once again, a big round of applause. Thank you so much to our Shaw laureates. And to commemorate this very special occasion, ladies and gentlemen, would like to now take a photo with everybody. So together with our scholars and our guest speakers and also with our audience, now with our photographer, please take a photo for us. Yeah. May our guests on that side please move to the middle. Please take your seats in the middle. So that we can have everybody sitting in the middle and we can take this photo together. Thank you very much for your cooperation. So filling some seats in the middle and at the back. We still have 2 more seats in here. You may want to take these 2 seats. And also on the last row, thank you very much. Okay, starting with our photographer in the middle. All right, big smile, big smile. Please face the cameraman on your right. One more. All right, do we need some video? What if we say hi to the video? Hi! Do we have some good photos already? All right, thank you very much, ladies and gentlemen. With that, we have now come to the end of this seminar. Thank you once again for joining us in person and through the live webinar today. Thank you very much. We hope you've enjoyed the program today, and we look forward to seeing you once again at the future Hong Kong Laureate Forum events. Thank you so much. Stay safe, take care, and goodbye.